Hey, Alan. Okay. So anyway, let's take a look. And so of the four, we're going to study three of them because they're polymers, and that's kind of what the last part of this chapter is about. And so let's go down here, and we found out that proteins, if you ask a biologist, they'll say they were the most important macromolecule. Ooh, macromolecule. Did you use that term when you took your... okay, macromolecule. Not little H2O, not little CO2. Even C6H12O6 is little compared to these guys. So anyway, some of these are called macromolecules, and proteins are polymers of what? What are the monomers? What are the building blocks of proteins? Amino acids. See that? Now, what I ask you to do is to pick, and some of you are very smart, you know which ones to pick. What I ask you to do is, you can do them in your book, or also have them here, and to pick 3 amino acids that are polar. And so you have to know how, and what I'll do is, I would give you, what I would end up doing is, I would give all of this to you, the basic structure of an amino acid, and then you would put the R group there, which that's the simplest R group you can have, and then you put glycine. And you said, yeah, yes, non-polar. And then the next one, I think, is the next simplest, and you don't have to pick this. You said, no, I want to challenge myself. Yeah, I'm going to do that one. Okay. But anyway, you would put this up there, and you'd say alanine. See that? And then I will do that one for extra credit. Okay. Let's see. What's another easy one? Anybody else see an easy one? I don't know. Maybe like that one, huh? You like this one, or the benzene ring, or I don't know. I will tell you that it's rare, but not that rare, that every baby, when they're born, they're actually tested for a thing called PKU, phenylketonuria. And so what happens is a certain number of humans, when they're born, they can't tolerate the amino acid called phenylalanine. And if it builds up in their body, it can actually cause their mental, normally they have a normal mind. It could actually cause a delay in their mental abilities. And so they want to test it because if it's something they can fix. They say, hey, just don't give the baby phenylalanine. Maybe when they get older, they'll be okay. So they have, they would have special artificial milk that it says contains no phenylalanine. And then the PKU babies would come up normal, like that. But because it's so easy to fix, that's why they want to test all the babies, even though it's not like, not like 1 in 10. I don't know numbers. 1 in, who knows? 1 in 5,000, 1 in 10,000 maybe. Anyway, so have you ever heard of phenylalanine before? Okay. You have is it from PKU?
Yeah.
Oh, okay. Yes.
Oh no, I was just saying that.
From PKU? How do you know anything about?
Last year in AP Bio, we had to do genetic diseases.
Ah, so you studied PKU.
Yeah.
Ah, excellent. Okay. Well, anyway, so the building blocks of proteins are amino acids. And so how many different kinds of proteins do you think there are? How many? So, actually, I don't know, but if you give me a guess, I'll say maybe. But it's millions. It's millions. But even though there are millions of different kinds of proteins, and some of the smallest proteins might be 60 or 70 amino acids long. And some of the long ones might be 570, 600 amino acids long. Those are different, aren't they? They're a lot different. And so how many combinations can you do? And by the way, how many amino acids are there? Only about 20. That's what's interesting. The building blocks, these amino acids, there's only about 20 of them, but how many ways can you arrange it? Huge numbers, right? Huge numbers. Okay, what is it? I'm sorry.
18 of those used in every protein?
Say it again?
Aren't 18 of those considered necessary amino acids?
18 what?
18 of those amino acids necessary or...
I don't know. I'm trying. I guess I don't know if I've ever read 18 you have to have and 2 you don't.
I thought it was like necessary.
If you read that somewhere, I'd like to read it myself. I've never read that, but maybe so.
I just read that there was a guy who tried to prove evidence life, so we're not, I'm not
Not
sure.
one of the 20s?
I don't know if that was a part of the
Oh.
20 or if that was,
Um,
which might explain the 18 number.
Without a doubt, when I was in high school and even in college, when I saw the Urey Miller experiment, as you're calling about,
Yeah.
it strengthened my atheism, and I was an atheist, and it said, see, I tell people, see, even in a flask with quote artificial lightning and what we think the environment was on the Earth, which we don't know really, and by the way, biologist books have changed it. The ancient Earth had this kind of atmosphere, and that's what happened. Blah blah blah. And now new evidence. Oh, the ancient Earth had this kind of atmosphere. We were wrong. They don't say we're wrong. But even though they've changed the idea of what they thought the ancient atmosphere was, they still kept that in the books. You know why? Because it's a great selling point for atheism. And certainly it worked for me. I mean, that was working for me. But if you really understand what a living thing is, the fact that you can make an amino acid, that's just saying, that's a building block of life. That's a building block of life. Now,
I mean, that's,
so if you can make an amino acid, then you can make that cell. Really, oh, that's a little jump, isn't it? Really? Think about a jump from an amino acid, 2 amino acids. They have to join together, and what sequence? And what are they going to do? Let's say you put them in sequence of 10 in a row. What are they doing? Nothing. What's your purpose? Nothing. I'm waiting, though. I'm waiting around for some purpose. And I used to not think about that. I don't know why. I thought I was smart. But why didn't I ever question that, you know? They said, if you can make an amino acid, you can make a life. Really? You know what you have to do to have life? Wow, it's unreal. It's unreal how much you have to have. Okay, well, anyway, I don't want to get too much in that, but certainly if you take biology in college, you will get that. It'll be shoved down your throat. Every chapter will have it in there. It won't talk about just the science of it. It'll talk about the idea of evolution. And again, you certainly don't want to tell people you don't believe in evolution, because I can do an experiment that can show evolution, because their definition is so wide. It's so wide, I can do microevolution. That happens all the time. But they want you to say that microevolution also means that macroevolution took place also. And so it's kind of a catch-22. You can't say I don't believe in evolution, because you'll get caught. And then you say you believe in evolution, see?
say that's in macroevolution.
Yeah, you can say I don't believe so. The macroevolution. I don't think the evidence is not there. All right. Well, let's go back. And so we have these amino acids, and why would and all amino acids have the same basic structure, don't they? Don't they have a central carbon? They always have a hydrogen down here, and then what's this end called? That's the COO, isn't it? What's that mean? Isn't that an organic acid? That's the acid end, and this is the amino. I get it. Amino acid. Good name. And the only thing that makes one amino acid different from another is this thing called an R group, and the simplest R group is H. What's the next simplest? CH3. Wouldn't that be the next simplest? That's about as simple as you can get. And then some of them get kind of complicated. All right. Now let's go to down here. How do you get 2 amino acids to join together? And what you have is, and again, we need enzymes. We already need a well-made protein that acts like an enzyme. So we need an enzyme that's going to grab a hold of an OH here and an H here, and now I want to form another bond, I want to form another bond, and now this CH-CN bond, that's called a peptide bond. So what we have here, I got up my little visual aid today. This is from my kids from a long time ago. My son is 42 years old, and he used this. And so when he got done and the other got done, but these little things here, I put little amino acids on it, isoleucine, and there's tryptophan, cysteine. Wait, so what happens is, in order to get these two to come together, they don't have any bonds. So somebody has to take an H off of you and an OH off of you. Which end gets the H taken off of it? The amino end or the acid end? Take a look.
Acid.
Oh, the acid end has what taken off? The OH. Or does it? So if I'm going to take off a water, I can't get any O's from the amino end, can I? So I take an H off the amino end and an OH off of the carboxylin, right? And so then they'll form a peptide bond. Okay, now, and if you remember your biology, DNA tells them how to do this by making a messenger RNA, and the messenger RNA says to the transfer RNA, go get one of these. Now go get one of these. Now go get one of these. And then what you do is you end up making a polypeptide chain. Isn't that right? No wonder they call it a polypeptide chain. Now, it's not a real protein yet because it hasn't twisted into all its different shapes. So what is the primary structure? What is the primary structure of an amino acid? What is it? Somebody said it. The sequence of amino acids. You're right. So, so we have. Whoops, wrong page. Ah, I gotta find it. It's in your book. So the sequence. I can't remember what page it is. Sorry. Here it is. Okay, so the sequence of the amino acids are the primary structure. And so if you have cysteine, alanine, isoleucine, that's your primary structure. Now, because each amino acid has a different R group, some parts of this chain are going to be attracted to other parts. And so the secondary structure, they said, and as right now, as far as you're not biology majors, all right, the only two I want you to know about secondary, what are the two things that can happen to a polypeptide chain to give it another structure? Somebody said it. What? What are the two kinds of secondary structures that the book talks about? Somebody said it. A helix, which means that. Did you say helix? I hate math. So maybe it could form like a little helix. See that? Like that. And that's one of them. What's another one? A pleated sheet, right? Now those are called secondary structures. And then after all that's done, there's a tertiary structure that says, okay, now, okay, now I got all this going, and then guess what? Now this part still is attracted over here. Okay, and so, and it makes that noise and everything. So maybe this will be the final structure of the protein. Okay, I've used these for a long time. But maybe that's going to be the final structure of the protein, and that's, as long as it keeps that structure, it works. But if you allow it to change pH or temperature very much, it'll come unwound and become denatured. And a denatured protein doesn't work. Yes?
So, something I've always kind of wondered is, don't cells often need specific proteins in specific parts of the cell?
Okay, now that's a whole other story about, here's a cell who makes a certain kind of protein. And then that protein has to get out of the cell, and then it has to get over somewhere else. And that's a whole other ball game. We could take that would take another class almost. But I think that's unreal how parts of the cell will package proteins. They'll package them into little sacs. And then the sac will go into the membrane, fuse into the membrane, and be sent out. That's one way to get it out. That's amazing to me.
But what tells the cell to do that?
Okay, I can say God, but okay, I can say that scientists think that the DNA is in charge of telling how to make a protein. Now, because the cell also has other things in there like endoplasmic reticulum and lysosomes, and because it has these other things, that the theory is, if you're not a believer, you would say, well, these get attracted over here to that sac. And then they get in the sac, and little fibers in the cell transport the sac to the edge, and then they release it. Why would they release it? Well, because these chemicals come in contact with these chemicals, and it's just unreal to me, I understand if you don't want to sit there and really figure out how a cell works, I can see where you think evolution just says, just like that. Oh, I'll make amino acids. Oh, I'll make a cell. Hey, if you don't understand unbelievable intricacies of the cell, then I could see you saying that might happen, but it's just not real. It's just so unreal. Everything has to happen. And the thing's microscopic. And yet it does all that stuff. And how do we even figure this out? How do we even figure out that this last medicine has something to do with messenger RNA? How do we figure that out? So like I said yesterday, I'm glad that some of you will probably go into science, and some of you will like the idea of trying to answer questions, and some of you might even become researchers. So we'll see. All right, and then so we have the tertiary structure. Means I'm folding in the final shape, and as long as I keep that shape, I'll work like I'm supposed to. In the 9th grade, you probably don't remember this, but you might remember, you didn't know what they meant, lock and key. Okay, so some biologists talk about the shape of that protein being so specific that when I come over here, and let's say this is sucrose, and I'm called sucrase, that means enzyme, doesn't it? My shape fits right here, and when I do that, there's a lot of attraction, boom, 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 and it breaks down sucrose. But if I run into something like glucose, nothing. It doesn't happen. It's almost like your key has to fit in this lock, and then it'll happen. And that's what they call that lock and key theory about enzymes like that. Okay. Anyway, if you go through here, you'll see that they talked about denaturing proteins and that kind of thing. But I think I'm about done with that. All right. There's one more thing that is really weird. There's something that's really weird, and you know what? I didn't make the model, so I'm going to have to do that tomorrow. I was going to talk about this thing here, and I'm going to wait on that since I didn't make a model for you. It's called chirality. But let's remind myself that I'm going to come back to it, okay? All right, what's the next one? What's the next macromolecule? Oh, I know. You know what I want to show you? Darn it. Now I lost my page. What was that page that hemoglobin was on? What is it? Here it is. Okay. What I think is really neat about hemoglobin is that you got this heme group, these heme groups, and yet they're all carbon, hydrogen, oxygen, nitrogen. Okay, organic. And then what's in the middle? Iron. And then you go down here, and here's chlorophyll, one of the most important chemicals in living plants. And you got to look, it's very similar. This is not similar, I get it. But this part's very similar. What's in the center of that grouping? And as somebody brought up, and I should have remembered it, crabs and Mr. Spock on Star Trek, what is found in the center of his heme groups? Copper. And that's why he has blue blood, supposedly. So anyway, if you want to read about that, I just want to show it to you. Now, another thing I want to show you is here's a final structure of a protein, and see how you can see little spirals here, like that. Anyway, that might be what a protein looks like. I don't want to show you one more here. What's this thing? It said here, it says hemoglobin dramatically demonstrates how sensitive the function of a biomolecule is to its structure. In certain people, and the synthesis of proteins needed for hemoglobin, if an improper amino acid is inserted into the protein in 2 places, this may not seem to be very serious, since there are several hundred amino acids in there. However, because the proper amino acid, the hemoglobin drastically, oh, here it is, I missed one. The incorrect amino acid was non-polar instead of polar. And that 2 amino acid difference out of a few hundred causes the red blood cell not to be at normal shape. And what do you call these kind of red blood cells? Sickle cell. And you probably had that in AP biology, didn't you? Even though sickle cell is much more prevalent in the black population, And if you knew somebody, does anybody know anybody who had sickle cell anemia? You do? Okay. Would you say that you've been taught about that? Have you been taught about it? Have you been taught about it? See? Now, there are other things that happen in the Caucasian population. It's called cystic fibrosis. Have you been talking about that? Have you been talking about that? Probably not. There's another one called Tay-Sachs. That's more prevalent in the Jewish population from Eastern Europe. There are different, that's genetics. I'm not here to teach you genetics, but yes.
So for those, like I'm thinking specifically of cystic fibrosis, like that is one nucleotide difference. So is the DNA telling, like,
Wow.
that's messing up the,
To talk about how sensitive that is, one nucleotide on DNA codes for the wrong amino acid. So that means one amino acid? Is that what it is?
I think so. So if it's that, is it then the DNA's fault or is it the amino acid's fault?
Okay, so any change in the sequence, and we haven't done DNA yet, the sequence of bases, that's called the genetic code.
Right.
The genetic code, the good news about genetic code is sometimes you can have an error and GGG might call for the same amino acid as GGA. So there's triplets.
Yeah.
Okay, so that's the good news. You can make little mistakes and get by with them. If you make the wrong mistake, that's called a mutation. Any change in the DNA is called a mutation. Now, we won't know there's a mutation if if one of those bases happens to code for the same amino acid anyway. Does everybody remember that in ninth grade, how many bases it takes to code for amino acid? And over 55 years ago, Watson and Crick won the Nobel Prize for this. Not 4. And they're called codons. Codons are 3 amino acids, 3 bases in a row on a messenger RNA. And those 3 bases, of course, were based with the DNA. I'll get the DNA. I just want to tell you about this, that there are some people that are lactose intolerant. That's also like I think it's one amino acid in the wrong place. And so that's how sensitive the structure has to be. You're making this protein, but it's not working. I'm making the thing that should digest milk, but it's off by a little bit and it won't do it. So they've had ingenious ways of fixing these. Are you guys up to date on any of this? Like, do you know what gene therapy is?
Yeah.
Do you know they actually are giving people things that will get into their cells? And I could even do it with a virus. Take a virus and said, hey, you're a good virus. The bad parts of you, I'm gonna get rid of that. I'm gonna put in a human gene, a human piece of DNA into the virus, and you're really good at affecting lung cells anyway. So here, give the patient this virus. And it goes in there, reproduces, and then all of a sudden you're putting this new piece of DNA into a cell that had a bad piece of DNA. Wow, that's like science fiction, isn't it? Now, it is going to lead, if we're not careful, it will lead to a very, very bad thing. If we can manipulate DNA, and we can, I hate to say this. Anybody know what CRISPR is? You guys talking about CRISPR? That's some scary stuff there. All around the world, you can kind of almost type in on a computer and tell the machine, I want this sequence of DNA. And
there's some amazing benefits to that.
yes, and you know what? There are amazing benefits to TNT, and we built roads and built tunnels, and yet in a war you can kill thousands of people. And so I agree, it's not the invention that's bad. It's the sinful nature of humans.
I'm not saying whether it's beneficial or negative. I'm just saying, like,
Yeah,
you'd be foolish to not look into that. Because
I know.
It's possible to
Man.
save lots of lives, but it can also be used negatively.
If I knew humans wouldn't act like humans, I'd be okay. But man, if you had this device and humans said, look what I can do for the good of people, if that's the only kind of humans there were, we'd be great. But there's another kind that says, how much money are you going to give me if I do this? What? A weapon? Oh, how much money? Oh, okay. My own lab? Oh, okay. I hate that. But it is part of the nature, and that's why I'm getting off the subject. I'm sorry. But it's interesting, but it's not DNA's fault, but it's a mutation. You asked the original question. It's a mutation in the DNA. All right, let's move on. Now, the next the next one I want to talk about, the next biological polymer is carbohydrates. Now, carbohydrates, they, instead of ending in "in" or "ase," if it's an enzyme, they might end in "ose." And so, have you ever heard of glucose? That's a sugar. Dextrose, that's glucose. Don't tell anybody. In the hospital, dextrose is glucose. But anyway, have you heard of sucrose? That's table sugar. Okay. Have you heard of fructose? If you have Karo syrup at your house, you ever have that? That's like pure like fructose. It's like a simple sugar. So what we see here is what are the building blocks of carbohydrates? And they're sugars. Okay, sugars are the building blocks of carbohydrates. So what the monomers, you can have a sugar. So here's a sugar, and then you could connect a sugar to another sugar. Now, one of these would be called a monosaccharide. Put them together, you get a what? Okay, so the most common monosaccharides are glucose, fructose, and what's the other one? Lactose. I think it's lactose. And all of them have the formula C6H12O6. They're isomers. Now, if you put them two together, you can get a double sugar or a disaccharide. Table sugar, the kind is your home, is fructose and glucose put together. Maltose is glucose and glucose put together. All right, so now let's take a look at the structure. And I need to go back to your book because it also has some really good pictures in it. Okay, they show the non-ring form of a sugar. I'd like to show the ring form, but here it is. Okay. All right, now, can everybody see this right here? Okay, so this is fructose, and I'm going to go to the next page. And so what we see here is these are some sugars. And what you want to do is, how can I get a sugar to want to attach to another sugar? So look down here. Stay and look down here. So here's C6H12O6, and here's C6H12O6. This happens to be glucose. This happens to be fructose. All right. Now, how do you get them to go together? They don't want to join together. So what are you going to call it? What kind of a polymerization is it? I'm getting some fancy names in here today.
Condensation.
It is. Condensation. Just like the amino acids came together, right? So where's my small molecule? They're going to take an OH off of one sugar and whoa. Uh-oh. It should only take an H. It shouldn't take, they're going to take an H off another. So you take an OH off of one sugar and an H off another, and that forms water, right? Here's a question I love asking on the test. What if you were to put 5 sugars together, 5 glucoses together? What would be the formula of the big molecule? Put 5 C6H12O6s together, and this is a good question for high-level people like you guys, high-level students. Put 5 glucoses together, and what would the final formula be? Can you at least try it? And said, oh, well, I don't think I'll get it right. Well, try it anyway. Try it anyway. Alex, you going to try it? Put 5 glucoses together. You already have the answer? Oh. Everybody got it? It's going to be a funny looking math problem, okay? What would 5 times glucose be?
30.
C30, H60, O30, is that right? But in order to put 5 glucoses together, you had to take away 1 water, 2 waters, 3 waters. How many? 4. So you have to subtract 4 H2Os. Isn't that a weird? Anyway, so the final answer would be take away 8 H's, take away 4 O's, and there we got it. C30, H52, O26. Isn't that pretty neat? So what if I ask you about 99 glucose together? How many waters would you take away? I'm sorry, I mean 100. Dang it, gave away the answer. What if I put 100 glucose together? How many waters are you going to take away? 99. Okay. All right, well, anyway, instead of making a monosaccharide, you would have a disaccharide. And we have a lot of those. But where's the polymer come in? And look at this right here. Imagine doing glucose, glucose, fructose, fructose, glucose, 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 and you make it 10,000 long, 10,000 sugars long. What would you call that? It wouldn't be a monosaccharide. It wouldn't be a disaccharide. It'd be a what? Polysaccharide. Now, we have simple names for that too. They're called starches. I remember when I first had to learn this back in college, I always remember, wait, polypeptide makes proteins. That was my memory trick. Polypeptide, proteins. Polysaccharides make starch. See? That was my memory trick to remember which one was which. Now, we have starches in plants, and they're all over the place. Starches in plants, all over the place, right? You take a root, pull a root out of the ground, break it open. Oh, look at that milky stuff. You ever seen that? That's such pure starch. Get that? And it's long chain. And people said, oh, I'm going to cut out sugar. I'm just going to eat bread. Bread is starch. What's starch? Sugar, 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 sugar. When I put that in my body, little enzymes go, and it cuts all the little sugars. You're not going to cut out sugar by eating starch, are you? Now, some people say, I'm going to cut out store-bought sugar. Okay, you can do that. I'm going to cut out fructose. You can do that. but don't say you're not eating sugar when you go and eat a potato, because a potato is almost 100% starch, isn't it? All right, what's neat, and I'm not going to get into this. If you take a biology class, you'll figure out that there are many, many kinds of carbohydrates. Now, plant starch is one. Does anybody happen to know what animal starch? I actually store this long chain amino carbohydrate in what form? Does anybody know what you call animal starch? Wow, how'd you know that? That's great. Glycogen. That's really good. So anyway, so you could actually go quite a while without eating, and your body will just release some glycogen, and then your body says, hey, I need some glucose. Okay. And it'll cut them up, and every cell can accept glucose. You get that? Okay. Says here, 10% of the mass of your liver is what? Glycogen. Wow, what's it? Thanks, liver. Do you know the liver also gets rid of a lot of poisons and things, you know? Okay. That's why, I don't know why people want to eat liver, but I don't know, some people like it. But what does it do? It's garbage. That's where all the poisons are stored and trying to take apart. They try to take them apart. All right. Anyway, I will talk about lipids briefly. Lipids are not polymers, but they are made of, if proteins, here's proteins. Proteins have, they have C, H, O, N, and sometimes sulfur, okay? And that's what you find in there. And as a matter of fact, when you say to people, hey, I'll give you a whole lot of bread and water, you'll not starve. But then you come back 6 months later and they're dying. Why are they dying when you give them all the bread and water they can eat? They're malnourished. But I'm giving them carbohydrates. But you know what carbohydrates don't have in them? Carbohydrates have C, H, and O. And what are they missing? Nitrogen. Humans need a source of nitrogen to make their own proteins. You get it? If you're only going to give them this and water, they're going to be lacking in nitrogen. and that's where God, he's not ironic or anything like that, but you live in an ocean of nitrogen, but you can't touch it. It's N2. It's triple bonded. So we have to get it by eating food. And anyway, so it's kind of, now, I will tell you that lipids have CHO, and by the way, carbohydrates always have a 2-to-1 ratio of hydrogen to oxygen, like C6, H12O6, see that? So carbohydrates have a 2-to-1 ratio, H to O. But lipids have CHO, but you might have 100 to 1 or 50 to 1. Very, very few oxygen, most of it's carbon and hydrogen. Well, let's go to the last one. And this one is nucleic acids. And what's found in those? CHON and P, phosphorus. Okay? All right. So let's go to your handout sheet. Let's see. Oh, I did do a little thing on lipids here. Let's go to the last one. All right. So, yes.
What kind of foods have nitrogen?
Proteins. Now, proteins for sure. Every amino acid has protein, right? And so amino acid chains are called proteins. So if you want to feed the world, you've got to give them a source of protein. Now, what now?
Just like meats?
Meat would be one. There are definitely people around the world trying to figure out how can I make a source of nitrogen that where I don't have to kill an animal, that kind of thing there. And some people are trying to make some kind of a meat substitute that has nitrogen in it. And maybe one of you will do that. But they're malnourished because they can't even make their own proteins. And muscles are proteins, aren't they? Enzymes are proteins. So you're not going to give them anything to make proteins with. They're going to die eventually. Now maybe they go eat insects. That's what I do. I go eat some insects. At least I'm going to have some nitrogen. I mean, really, if you knew that all you had was milk, water and sugar, I'd go eat earthworms. I'd eat things like that. All right, well, let's do nucleic acids. And by the way, I didn't know in the 9th grade. It's very possible in 9th grade you didn't know what Na stood for. DNA, RNA. What's the Na stand for? Nucleic acids. And they first discovered in the late 1800s when they started doing staining cells. Somebody figured out how to stain cells. And one of the stains would stain mitochondria. And then one of them would stain the nucleus. And one of them would stain something in the nucleus. And so they said, I don't know what that is, but there's something in that nucleus. And they tested it and said it has some kind of an acid property. What are they going to call it? A nucleic acid. And that's how they came up with the name. Now, nucleic acids are polymers, but the building block is complicated. What's the monomer of a nucleic acid? It's this great big thing here. It's called a nucleotide. Now, nucleotide is a sugar phosphate base. Sugar phosphate base. Sugar phosphate base. Get it? So if you have all three of those, that's one nucleotide. Now I need about 3 million of those. And then I can make maybe DNA. Now let's take a look. What sugar is found in DNA? Deoxyribose sugar. Okay, let's take a look. Now, I'm not an expert on history or chemistry, I'm not. But I believe this sugar was probably discovered before that one. Because this one, they gave it, it's a 5-carbon sugar with a O in the ring, 5-carbon sugar, and somebody said, I'm gonna call it ribose. Okay. Then they found another one. This is almost like ribose, except it's missing an oxygen. Let's call it George. No. They said, no, it's just like ribose, except it's like one oxygen missing, that's all. What do you want to call it? Deoxyribose. I get it. Ribose? Deoxyribose. Okay, so if you want to make DNA, I need deoxyribose sugar. You want to make RNA? Use this sugar, ribose. I get it. What about the phosphate groups? They're all the same. All the same. Everybody's phosphate group's the same. What else is different? They have these little bases. Now these bases sitting here, and your book has all of them right here. Oh, here they are. Wait, remember in biology class, A, T, C, and G? Remember that? Okay, adenine, guanine, thymine, cytosines, remember those? Those are bases, and because they have nitrogen in them, they're called nitrogenous bases. Okay, so they have these nitrogenous bases, and what are they found in DNA? DNA has A, T, C, and G. I didn't do this, but RNA almost has A, T, C, and G, except there's no thymine. That's so weird. But there's a chemical that's almost identical to thymine, and it's called uracil. And so are you. And so are you. But anyway, it's called uracil. And so it's almost the same basis, except there's no thymine. Uracil kind of acts like thymine, your RNA. Now, these are bases, okay? And what happens is sometimes you get one part of DNA right next to another part of DNA, maybe at one strand, two strands, and what holds them together? And here's an A sticking out, this little base, and over here's a T. And A base pairs with T, hydrogen bonding. And what are the other pair? A with T and T with A. And you know how I memorized this? When I came here and I was teaching biology, A&T University, UNCG, or Guilford College. Okay, so C base pairs with G, G with C, A with T, T with A. So when you look at DNA, if you see a adenine sticking out here, you'll have a thymine over here like that. And so if you show this, this next picture, whoops, no, she didn't put one. I gotta go over here. Now, and I don't have 2 more, give me 2 more minutes, okay? All right, here's my nucleotide. Watch, sugar, phosphate, base. Everybody get it? That's one nucleotide. Now, what I did not understand when I was in college was I saw pictures like this. Whoa, hey. And I even saw where they untwisted it and make it look like a ladder. You see that? But I didn't understand what they meant. And what it is, guess what's right there? What's right there is a sugar. And then there's a phosphate, and a sugar, and a phosphate, and a sugar, and a phosphate, sugar, and phosphate. So the strands of DNA and RNA, they're made of sugar, phosphate, very, very, very strong bonds. And then what's sticking off of the sugar? What's sticking off the sugar? The base. A, T, A, G, 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 A, G, G, A, G. See that? And that is the sequence of bases. That is called the genetic code. The genetic code, what made you different than me, is we have the same number of chromosomes, same size chromosomes, but the sequence of your bases is different than the sequence of my bases. A lot of your sequences are the same as me. You have 2 eyes? I do. See? We have a lot of things that are very similar. Some are not similar. And so that's pretty neat how God made this. You know, I think about CDs. And if a CD, all it is a shiny thing where it's shiny and then they burned a hole. They burned a little pit in it. And then the pit, shiny. And if I say it's too fast, I'll say the wrong word. But pit, shiny, pit, shiny, pit. I'm careful. And so what happens is there's only 2 letters of the alphabet in a CD. It's like shiny or not shiny. 1, 0. But how many combinations can you make? You pick up 1,000 CDs and they all have different music, don't they? There's only 4 letters of the alphabet in DNA, A, T, C, and G, and look what it makes. It makes all of us. It makes all those trees. It makes the flies. It makes everything. An old movie, E.T. A lot of people, if you watch it, the one thing they said, remember how they wanted to poke him and all that? He's DNA. It's not DNA. He's from far away, but he's DNA. And they love that. They love that kind of stuff. You know where they're going to Mars right now? You know what they want? You know what they want to find? They want to find DNA. They'd love to find DNA.
They expect you to find DNA?
You know what the hard thing is it's very difficult to go there. And if they find DNA, it's very difficult for them to say, and I promise you, nobody on earth contaminated that. That's not, that's going to be hard for them to say that it wasn't something contamination. It could be bacteria in there. And that's hard. When you go to another planet, how do you know you didn't take something with you? Yeah. Okay.
Attention to young students, student drivers.
All right.
Student athletes.
Tomorrow, the quiz, okay. One side has the isomers of octane. Structure, you put the structure and name. And then I have like 3 other things you have to name. And on the back, you give me the 3 amino acids that are polar, 3 that are non-polar, and that's it. That should be a nice grade for you because I'm telling you pretty much what the quiz looks like.
For the other names,
What now?
for the other ones that we have to name, will those be like the functional groups? Because we're
Okay,
learning
it'd be
those on
like the
Thursday.
last quiz where I made remember how I made a structure and it was like 2-ethyl-4-pentene or something like that. This time I might ask ones like maybe alcohol. Like,
Okay.
you know how to name alcohol?
So do review the functional group names?
Yeah.
Okay.
But there's only like, I think you have 4 questions there. So 18 isomers, 4 questions, and then the amino acids on the back.
Okay,
Thank you,
thank
Mr.
you.
Greeny.
Okay. All right, bye.